Well, I'd first like to thank the Dean, the department, and especially the foundation for developing these professorships. I'm really honored to be a member of the inaugural class of what I hope are many other MSU Foundation professors. It's a wonderful thing, both for on campus and for our visibility off campus. In thinking about what to talk about today, I just have a handful of slides, and I just sort of want to cover at a high view the topics that we've been working on in the lab and really what drives us. And I guess if I had to come up with a theme, it's really we work at the interface of plant biochemistry and human health. And so that's really what drives everything in the lab. On the right here, these organisms we've used to pick apart vitamin E and provitamin A or beta-carotene synthesis with the idea of being able to manipulate it to increase the levels of those vitamins and others in crops. And I'll talk a little bit more about that in a second. This here is a Camptotheca acuminata tree. This is a Chinese happy tree. I don't know why they call it that. It seems no happier than any other plants. But it makes a really important anti-cancer chemotherapeutic. And you may not recognize these structures, but you hopefully recognize these names: beta-carotene and alpha-tocopherol, provitamin A, and vitamin E. And this guy, camptothecin, no one but me really recognizes. So before I tell you just a few slides of each of them, I just want to give you the big picture. And I've been putting this up probably for 2 decades. This is the number of compounds that have been characterized in nature. And as you see, the bulk of them come from plants. So plants are really the kings of metabolic diversity on the planet. And I estimate at least 1 million here, but this used to be 200,000. Every time, John O'Rourke's my canary in the coal mine for this. Every time he gets comfortable, I double the number. So we just had another doubling, John. So, but with 80,000 or 1 million compounds to work on, why would I just sit on vitamins for these 12 or 14 compounds for the past 20 years? But before I tell you that, I guess one more thing, I forgot this. What's really driven a lot of the work in our lab is this slide here. And so I realized, I pulled this out of a talk from last year, that this almost covers all of my career at MSU. I started in 2000. The cost of generating 1 million base pairs of DNA sequence was $10,000. In that intervening 15 years, it's come down to less than a penny, right? So this is a disruptive technology, right? Hopefully you only have to live through these once or twice in your career, because it's a wild ride. This could also be my hair loss, I think. What it's enabled is some approaches that we really couldn't even think about 10 or 15 years ago. And I'll tell you about this MPC, this Medicinal Plant Consortium, in a little bit. That's the happy tree and why we're working on it. But then this other approach, GWAS, this is genome-wide association studies. What this is, you can take thousands of individuals and phenotype them for a trait, baldness or vitamins, and then you can put tens of millions of markers onto their genomes, and then you ask the question, which of those tens of millions are associated with the trait that I'm interested in? And once you have that piece of DNA, you can ask what gene is nearby. Right? So that's something that really hasn't been possible. So why do we work on vitamins and continue to? It's because of this slide. And you all know these numbers. Sometime last decade, we passed 7 billion. Most of those people are in underdeveloped countries. This number, under nutrition, that is, people getting less caloric intake than they require each day, this number is actually held steady for the past 40 to 50 years. And that's because the Green Revolution has allowed us to grow more and more food on the same acreage. So we've been able to provide the quantity of food to really drop this number. It used to be 1 in 3.5, and now it's down to 1 in 7. This number has been stubbornly constant the past 50 to 60 years, and maybe even going up now. And the reason for that is, although we're able to grow much of the food that we need, the quality of it has really not been addressed. And so that's really what's driven me to focus on vitamin pathways. We like to know what they do in plants, but we also recognize that probably the biggest impact we could have is out there in the real world. And so the reason we stay on this is because I challenge you to think of another problem, another test in life where if you get 10% right, you can affect 200 million people, right? So it's one of those things that even if you make a tiny movement, you have a big effect. So the reason for that micronutrient malnutrition being so pervasive is, again, we're a victim of our own success. And so what I've done on this slide is to calculate for you the fold increase for any one of these nutrients here that you need to make in order to get your RDA in a kilogram of boiled plant products. So for us, eating 2 pounds of boiled maize flour or 2 pounds of wheat mush a day sounds really awful, right? But it's actually a good day for more than half of the planet, right? So you can see the problem with the source of micronutrient malnutrition is the guys in green here are compounds we'd have to increase twofold. You can do that just by selecting different varieties, right? But for things that you have to increase several fold to several hundred-fold, you really need directed breeding, probably genetic engineering, and most likely a combination of the two, right? So what we've been focused on is identifying and understanding how all these compounds are made, and then how to manipulate them in plants. And so this GWAS, genome-wide association studies, allows us to probe the whole genome, 30,000 genes. We'll just talk about the 100 that we know of that are potentially involved in making these two compounds. Now, if I had to go in and do experiments on those 100, I need 2 or 3 more careers to get through them, right? So what we really want to do is identify the important things that we can focus on and put more effort in. So taking this approach, and we'll ignore all the math behind it, but of the 100 genes that are potentially involved in these two pathways, only 5 of them are really what I would call key for determining beta-carotene, and this is in maize seed. 6 of them are key for determining alpha-tocopherol or vitamin E. This gives us a list of genes then that breeders can go in and manipulate, and we also learn a lot about whether they can play well together, right? Some genes don't really behave nicely when they're paired up with each other. So the efforts really in the next several years are going to be to try to develop similar levels of understanding for the rest of these guys highlighted in yellow. And this is a grant that I should be writing right now instead of talking to you guys. But that'll come this afternoon. So I just want to turn the last couple slides to this medicinal plant consortium that we had funded through 2009 to 2012. And this was a part of the Recovery Act, funds from the Recovery Act. And really, being at MSU really enabled me to participate in this to the level that I did. MSU really led both the metabolite analysis and the transcriptome, the DNA sequence analysis for this entire group. And I think being at MSU, people actually look to us for leading these types of efforts. And so the general approach of it was just we recognize that plants have been really the source of over half of the medicinal compounds that are used now currently in human health. And there's a ton of things out there that remain to be discovered. From my perspective, there's some really interesting biochemistry to play with, but again, in learning it, we can actually potentially impact human health. And so what this large consortium did was to develop transcriptomes, and this is sort of the readout of the genome for making all the proteins that some of which are involved in making the compounds we're interested, and the metabolome, all the different metabolites that are made by these different tissues. And this is, again, available worldwide. A number of groups are using it to pick apart pathways in these 14 different lines, plant lines. So what we've been focusing on is this compound here, again, camptothecin. Hopefully you guys never actually need to use it, right? But it's a very potent chemotherapeutic. If you're unfortunate enough to come down with one or more of these cancers, it's really one of the frontline chemotherapeutics for them. And there's semi-synthetic derivatives that the chemists have made. You can recognize a sort of chemical thing here of camptothecin in both of these. From my perspective, we were interested in understanding how it's made so we can manipulate it and possibly make some derivatives in plants. And so here's our Chinese happy tree. 2 of these other plants that we chose for the Medicinal Plant Consortium also make this class of compounds. So they've been studied a bit more than Camptotheca, so we can use information from here to go in and identify candidate genes in Camptotheca for the synthesis of this compound. This one up here you guys should recognize. This is Catharanthus roseus, It's Madagascar periwinkle. Home Depot sells 1 billion of these guys every year in the spring. You'll see them everywhere, and nothing attacks them in your garden, and it's because they make this chemotherapeutic, which is toxic to bugs as well. Okay? So being at MSU, really, we wouldn't have gotten this project up and running, and I show you just this last data slide. This is a picture of the greenhouse about a month ago. We have a greenhouse now of transgenic Camptotheca trees. I think we're the only lab on the planet that's been able to transform this thing. And we're trying to pick apart the pathway, so the way we do it is we go in and break it. So we're actually causing these trees to make less camptothecin, and that tells us that gene that we've modified is actually involved in the pathway, right? So this is fun, and I can guarantee you this wouldn't have happened without being here. So I don't have a lot of pictures to show, but I wanted to put up just the list of people. And again, as Christoph said, you know, we don't actually do any work. I'm sort of like the Scarecrow in The Wizard of Oz, right? They gave me a brain, I sit in my office and think deep thoughts, right? But it's these folks that actually make the science happen. And so in yellow are the current labbies, past MSU labbies here. Some of you will recognize their names. In asterisks are undergrads, and we always have sort of between 3 to 8 undergrads in the lab at any given time. I try to select, when possible, undergrads who they're the first person in their family to go to college, because I was the same, and working in a lab was pivotal for what I'm actually doing now. Lots of collaborators, and this is one of the great things of being here at MSU. There are many, many people to work with expertise that complements your own. And it gives you a platform on which to go out and interact with the many people around the globe that we need to do these investigations that we undertake. So with that, I'll just close and again just thank the foundation and the administration for selecting me and Christoph and the others for this inaugural foundation professorships. And thank you all for your time.